Imagine for a moment that you have 18 perfectly engineered genetic cures. You mix them all into a single syringe, inject it into a patient, and suddenly realize you have you've completely scrambled the targeting system for the entire brain. Cures meant for the motor cortex are suddenly firing in the striatum. The targeting is just, utterly corrupted.
Yeah, it is basically the absolute nightmare scenario for genetic engineering. I mean, we pride ourselves on precision, right? But biology has a way of introducing just absolute chaos the moment you try to take a shortcut.
Right. And welcome to today's deep dive. We're looking at an incredibly dense, frankly mind-bending 2026 Nature Communications paper from the Allen Institute. Our mission today is to investigate the hidden chaos of what they call multiplexing in genetic screening. Which
is fascinating because if you're working in big data, biotech, or honestly, if you're just fascinated by the limits of modern science, this paper is going to fundamentally change how you view these high-throughput shortcuts.
It really is. So to ground this for you listening, let's start with the basic delivery mechanism here, which is enhancer AAVs.
Right. So if you're designing a gene therapy, the AAV, that's the adeno-associated virus, that is your delivery truck.
It's the vehicle.
Exactly. It's a hollowed-out viral shell, and it's carrying your therapeutic DNA payload. But a truck obviously needs an address, right? And that is where enhancers come in.
Right, because you don't want the payload going everywhere.
Exactly. Enhancers are non-coding DNA elements that act like highly specific genetic zip codes. They interact directly with the host cell's transcription factors to dictate exactly which cell type turns on the payload.
So we are talking pinpoint accuracy here, like targeting just astrocytes or just oligodendrocytes or, highly specific subsets of neurons.
Precisely. But the bottleneck in the field is actually finding the right zip code.
Yeah.
Historically, validating these enhancers is just agonizingly slow.
Because it's a one-by-one process, right?
It is. You clone one single enhancer, you put it on one AAV, inject one mouse, wait, and then slice the brain to see where it lights up. It requires immense time, money, and animal resources.
Which is why the researchers in this paper attempted what seems like a brilliantly obvious shortcut.
Yeah, the shortcut is called multiplexing. So the Allen Institute researchers took 18 highly validated enhancer AAVs. And to be clear, we know for a fact these specific enhancers work.
Like they are gold standard.
Exactly. When tested individually, one by one, they hit their exact target cells with over 95% specificity. So the big idea was, why not pool all 18 together, inject a single mouse, and read the results simultaneously?
It sounds so incredibly efficient, but wait, how do you tell them apart if they're all mixed together?
Right. So to tell them apart, they attached tiny 8-base pair genetic barcodes, they call these MBCs or messenger RNA barcodes, to each specific enhancer. They plan to use single-cell RNA sequencing to essentially read these tiny barcodes and map everything out.
It sounds like high-throughput biology at its absolute finest. But, and here is the massive spoiler for you, the experiment completely failed. When they mixed these 18 highly accurate AAVs together, the specificity plummeted to under 50%.
Which is just a catastrophic drop. A precision targeting system turned into overwhelming biological noise.
So the rest of our time today on this deep dive is going to be a forensic investigation into exactly why the shortcut failed. And let's start with the very first hurdle they hit, which was honestly just trying to extract the data. I mean, how do you even measure a tiny 8-base pair barcode inside a complex brain cell?
Right, so it all begins with the tissue preparation phase for the 10x Genomics RNA sequencing platform. The researchers were focusing on two specific brain regions, the motor cortex, which we call ALM, and the striatum, or STR.
Okay, ALM and STR. Yeah. Got it.
And they had to decide how to extract the genetic material from the tissue, so they tried two standard methods. Method 1, isolate the whole, intact cell. Method 2, chemically dissolve the outer cell membrane and just isolate the single nuclei.
And the data from that choice was shocking, wasn't it? Because over 90% of the whole cells they isolated clearly showed the AAV barcodes. But when they looked at just the isolated nuclei,
fewer than 10% had any detectable barcodes.
Which is wild. Why is there such a massive discrepancy between the whole cell and the nucleus?
It comes down to the physical life cycle of messenger RNA. The 10x sequencing relies on capturing mRNA. And what the researchers quickly realized is that the mRNA carrying these specific barcodes was incredibly efficient at escaping the nucleus.
It was slipping right out.
Exactly. The very moment it was transcribed, it migrated out into the surrounding cytoplasm.
So if you only grab the nucleus, you completely miss the barcode. Now, the researchers didn't just throw their hands up and accept this, right? They threw the absolute biochemical kitchen sink at trying to trap those barcodes inside the nucleus. And I want to walk through exactly how they tried to do this because it reveals so much about cellular mechanics. First, they tried modifying the genetic vector itself by removing an element called WPRE3. What exactly is that?
Yeah, so WPRE3 stands for the woodchuck hepatitis virus post-transcriptional regulatory element.
Wow, quite a mouthful.
It is, yeah. But basically, it's a sequence commonly added to AAVs because it acts almost like a biological exit visa. It actively binds to specific nuclear export proteins, aggressively promoting the transport of the RNA out of the nucleus and then stabilizing it once it reaches the cytoplasm.
Oh, I see. So the researchers hypothesized, hey, if we just delete this WPRE3 exit visa, the RNA won't be able to leave the nucleus.
Which makes perfect logical sense on paper. But in reality, it completely backfired. Removing WPRE3 actually reduced the barcode detection even further.
Wait, why? If it can't leave, shouldn't there be more of it in the nucleus?
You would think so, but biology is rarely a single variable equation. WPRE3 doesn't just export the RNA, it physically stabilizes the transcript. So without it, the RNA didn't just stay in the nucleus, it rapidly degraded. The signal just completely vanished.
Oh, wow. Okay, so genetic deletion failed. Then they tried physical interventions. They thought, maybe the temperature changes during the tissue prep are causing the delicate nuclear membranes to burst, physically leaking the RNA. So they rigorously minimized the freeze-thaw cycles. Did that help at all?
Not at all. The temperature protocol changes made zero difference. The RNA was actively escaping, not just passively leaking through broken membranes.
Okay, so then they turned to structural biology, and this is where it gets really clever. They engineered the barcodes with genetic nuclear retention motifs.
Right, these are specific sequences naturally designed to essentially anchor RNA to the internal infrastructure of the nucleus.
Right, so they tried the U7 scaffold system.
Yeah, the U7 small nuclear RNA naturally binds to specific proteins to form a complex, and that complex stays permanently in the nucleus to assist with splicing. So they tried attaching that scaffold directly to the barcode, hoping it would hold it in place.
And they also tried BORG LNCRNA motifs, right? I know LNCRNA stands for long non-coding RNA.
Yes, and these specific Borg motifs physically tether themselves to the nuclear matrix, which is like the internal protein scaffolding of the nucleus itself.
Like tying a boat to a dock? And finally, they tried sirloin elements.
Yeah, sirloin elements are sequences heavily enriched in what are called ALU repeats. These repeats are specifically recognized by nuclear proteins that essentially drag the RNA back into the nucleus if it ever tries to escape.
And despite all of these incredibly sophisticated biological anchors, the scaffolds, the tethers, the sirloin elements, none of it worked. Nothing boosted detection past 50%. They completely had to abandon the nuclear prep method.
Yeah, they had no choice.
To visualize this for you, imagine the cell's nucleus is a high-security bank vault, and these 8 base pair barcodes are tiny little cash withdrawal receipts. The moment the receipts are printed, they are instantly shoved under the heavy vault door and out into the main bank lobby, which represents the cytoplasm.
I love that analogy.
Right, so if your sequencing method only searches the vault, the nuclear prep, you find almost nothing no matter how much you try to genetically superglue the receipts to the floor. You absolutely must search the entire bank lobby, the whole cell prep, to find your barcodes.
That is exactly it. You have to capture the cytoplasm. But here is the truly terrifying part of this paper. Even when they switched to whole cells and successfully captured the cytoplasm, the data was still a complete disaster.
Okay, before we look at why the whole cells were a mess, let's just make sure we are locking this all in. I want to do a quick sanity check, a little mini recap. So if you're listening and taking notes, here are the first 3 biological realities to remember. Bullet 1. Enhancer AAVs are genetic zip codes that tell a delivery virus which specific brain cell to turn on.
Right. Bullet 2. Multiplexing them, pooling them together to save time, introduces massive noise and drops their targeting specificity way below 50%.
And bullet 3. The mRNA barcodes physically escape the cell nucleus. This means whole cell sequencing is mandatory because all attempts to biologically trap the barcodes, whether by removing the WPRE3 export signal or adding structural Borg and sirloin anchors, completely failed.
Exactly. So now we are using whole cells. We can finally see the barcodes clearly, But the data makes absolutely no sense. The targeting is completely scrambled.
Yeah, let's talk about that. Give me an example of what they saw.
Well, for instance, there's an enhancer called AE07709 meter. We know for an absolute fact it strictly targets the striatum, but suddenly in this multiplex pool, its barcode is heavily lighting up in the motor cortex.
It's like a zip code for a neighborhood in New York successfully delivering a package to a house in Los Angeles. How is that physically possible?
Right. It forces us to look backward, away from the brain, and into the manufacturing process itself. To understand why the targeting scrambled, we have to look at the biological factories used to actually build these AAVs, the HEK293T cells.
Okay, so how did they build them initially?
They initially used a standard pooled AAV packaging method. They put all 18 different DNA plasmids, each containing a specific enhancer and its matching barcode, into a single massive bath of these HEK293T producer cells.
Wait, let me visualize this. It's like an auto factory trying to build 18 entirely different models of cars on the exact same assembly line simultaneously. You've got parts for sports cars and heavy-duty pickup trucks all just thrown onto the same conveyor belt.
That's a great way to think about it. And inside the living cell, that conveyor belt is driven by DNA replication and repair machinery. When you flood a single cell with 18 highly similar homologous DNA plasmids, the cell just gets confused.
Confused how?
Well, as the DNA is being replicated, the strands can occasionally break. The cell tries to repair them using a process called homologous recombination. It physically crosses the strands over, seamlessly joining the enhancer of one plasmid with the barcode of a completely different plasmid.
Oh, wow. So to catch this happening, I know the researchers used a technology called Oxford nanopore long-read sequencing. And I really want to clarify why this specific technology is so important here. Because standard sequencing, like Illumina, is short-read, right?
Right, Illumina chops the DNA into tiny, like 150 base pair fragments.
So if your enhancer and your barcode are separated by, say, 2,000 base pairs, short-read sequencing literally cannot tell you if they're on the same continuous molecule. You're just looking at disconnected puzzle pieces. But Oxford nanopore is different.
Very different. Oxford nanopore literally pulls the entire unbroken strand of the AAV genome through a microscopic protein pore. It measures the tiny changes in electrical current as the DNA passes through, which allows you to read the entire intact sequence from end to end.
So they could see the whole car, not just a pile of parts.
Exactly. This allowed them to see exactly which enhancer was physically tethered to which barcode inside the viral shell.
And what they found is just jaw-dropping. Roughly 37% of the AAVs had discordant pairs. The biological factory had actively swapped the labels. The enhancer meant for the striatum was now physically fused to the barcode, meant for the motor cortex.
Yeah, this phenomenon is known as packaging chimerism. You created a chimera. The biological machinery took two distinct genetic entities and permanently fused them.
So to use the factory analogy, 37% of the time, the factory literally welded the pickup truck tires onto the sports car. That is a completely unacceptable error rate. So, obvious question. How do you fix a confused biological factory?
You have to completely segregate your assembly lines. You can't pool them at the plasmid stage. The researchers shifted to a mixed individual preps method.
Meaning they separated everything.
Right. They packaged every single AAV individually, one specific enhancer plasmid per batch of HEK293T cells. They let the virus fully form and seal itself completely inside its protein capsid, and then, only then, did they mix the 18 sealed viruses together.
And it worked. The Oxford nanopore sequencing showed that the chimerism rate plummeted from 37% down to just 2%. The labels finally matched the packages.
But, and here is where the paper gets truly fascinating, even with the chimerism practically eliminated, the specificity of the AAVs was still unacceptably low.
Which is wild. Fixing the factory did not fix the noise.
Exactly. The noise persisted.
Okay, let's pause for a second quick summary before we dive into why the noise persisted despite fixing the factory. So, tracking our progress. Bullet point 1. Pooling plasmids during AAV manufacturing causes the DNA to recombine inside the producer cells.
Bullet point 2. This homologous recombination causes chimerism, resulting in a massive 37% barcode mix-up where your genetic labels are completely scrambled.
And bullet point 3. To fix this, you must package AAVs individually in completely separate vats before mixing them into a pool. But, as we just discovered, even perfectly packaged AAVs still result in massive biological noise.
Which forces us to ask the big question. If the factory didn't mix up the labels, and the right zip code is firmly attached to the right package, why is an enhancer still lighting up a cell it's not supposed to target?
And this brings us to the deepest, most complex biological secret uncovered in this research. It's about what happens after the AAV successfully parks inside the target cell's nucleus. I always pictured AAVs entering the nucleus and just floating around independently, like separate, isolated little bubbles of DNA.
Oh yeah, most people do. But that is not what happens at all. The AAV genome is a single-stranded piece of DNA, and it's flanked by these structures called inverted terminal repeats, or ITRs.
Right.
When they enter the nucleus, they become double-stranded. But the cell's natural DNA repair machinery recognizes the open ends of these viral genomes as broken DNA, which the cell views as a massive crisis, similar to radiation damage.
So the cell panics.
Basically, yeah. So an enzyme complex aggressively sews these open ends together. The AAVs link end-to-end to form massive continuous chains called concatenamers.
So the individual delivery trucks are physically welding their bumpers together once they park inside the nucleus.
Exactly. And because they are now physically linked on the exact same continuous DNA strand, they start heavily influencing each other's regulatory behavior.
Oh, this is where it gets messy.
Very messy. Let's say you have an active enhancer like AE0140, which is specifically targeting parvalbumin interneurons.
Okay.
And right next to it, stitched onto the exact same chain, is a completely silent enhancer like AE0743 meters, which is only supposed to fire in striatal cholinergic neurons. Because they are physically touching, the active enhancer can accidentally force the silent enhancer to turn on.
Ah. And this is called transcriptional crosstalk. I think of it like a cheap, incredibly thin-walled apartment building.
Oh, that's a good one.
Right. So the AAVs linking together into concatenamers is like sticking a bunch of random strangers into adjacent apartments. One neighbor decides to throw a massive party and blast their stereo, that is your active enhancer. It is so loud that it aggressively vibrates the walls of the silent neighbor's apartment. To any sequencing machine listening from the outside, it looks like both apartments are playing music, even though the second neighbor never even touched their stereo.
That is precisely the mechanism. The intense local transcriptional activity forces the silent sequence to blindly produce its mRNA barcode. So your sequencing data then registers the silent enhancer as active. It's a massive false positive that entirely corrupts your targeting data.
Okay, so knowing this is happening, the researchers tried to stop the apartments from being built in the first place. They turned to in vivo testing using a very specific animal model, the SCID mouse. Now, SCID stands for severe combined immunodeficiency. I understand using SCID mice for immune studies, obviously, but why use them to fix a genetic crosstalk problem?
It's because of the specific genetic mutation that SCID mice carry. They naturally lack a crucial DNA double-strand break repair enzyme called PIDC.
Oh, I see.
Yeah, this is the exact enzyme responsible for non-homologous end joining, which is the process that actually sews the AAVs together into those long concatenamer chains. The hypothesis was actually brilliant. If you remove the PIDC enzyme, the AAVs literally can't form concatenamers. No chains, no thin-walled apartments, no crosstalk.
That seems like a silver bullet. You knock out the welding machine, the trucks can't weld together. Did it work?
Well, it was a wildly mixed bag. For certain cell populations, the targeting specificity did improve. For example, the striatal medium spiny neurons showed much cleaner signals. But for others, like oligodendrocytes targeted by the AA0410 meter enhancer, the specificity actually got significantly worse.
Wait, hold on. If you stop the crosstalk, how could the targeting possibly get worse? That feels entirely contradictory.
It does, but it highlights a double-edged sword in virology. Forming those concatenamers, stitching together into chains, actually helps stabilize the viral genome and massively boosts the overall gene expression.
Oh, so by preventing the concatenamers, the total brightness or expression level of the true recorders plummeted?
Exactly. The true biological signal became so incredibly weak that it was completely swallowed up by the background ambient RNA noise of the cell.
Ah, so you got rid of the false positives, but you also practically muted the true positives. So SCID mice are not a universal fix. What about a genetic fix? Because I know they tried something called insulators. What exactly is an insulator biologically?
So an insulator is a specialized sequence of DNA that recruits specific proteins, like CTCF. These proteins physically pinch the DNA into distinct spatial loops, acting as a structural and regulatory boundary.
Okay, so extending our analogy, it is literally like installing heavy acoustic soundproofing inside the walls of the apartment building.
Exactly right. They inserted the HSA2 and HSA4 insulators directly between the AAV components to physically block the loud active enhancer from vibrating the silent one.
And the results here are fascinating because they really prove how incredibly context-dependent the brain is. Adding the 5-foot HSA2 insulator worked beautifully in the striatum. It dropped the crosstalk noise from about 23% down to just 6%. But when they looked at the motor cortex,
it completely failed. The crosstalk remained stubbornly over 93%.
so the exact same genetic soundproofing worked perfectly in one cellular neighborhood, but failed entirely in another.
Which tells us that different brain regions and different cell types process viral genomes, regulatory proteins, and physical DNA looping in completely different ways. You absolutely cannot assume one genetic tool works uniformly across the brain.
Okay, let's do our third quick recap to make sure this is all tracking. So,
Right.
summarizing the biology of crosstalk, bullet point 1. Once inside the nucleus, AAV single strands are recognized as broken DNA and physically sewn together into continuous chains called concatenamers.
Right. Bullet point 2. These linked AAVs cause transcriptional crosstalk. A loud, transcriptionally active enhancer inadvertently triggers the silent barcode of its physical neighbor.
And bullet point 3. The clever fixes don't universally work. Using SCID mice to remove the PADC joining enzyme drops the signal strength too much, and adding genetic insulators only worked in very specific brain regions. The biological context dictates everything.
Right. So mechanically, we are trapped. We can't trap the barcodes in the nucleus. We can't pool the manufacturing without scrambling the labels. And we can't stop the viruses from linking up and crosstalking inside the cell.
Which brings us to the final, somewhat desperate attempt to salvage multiplexing in this paper. A system called SQUARE, that's spelled SCQER, standing for Single-Cell Quantitative Expression Reporters. If we can't fix the biology, can we somehow just filter the data? What exactly is this SQUARE system doing?
This is a purely computational, data-driven error correction strategy. Instead of relying on just one mRNA barcode, the researchers engineer the AAV to produce two entirely different barcodes simultaneously. One is the standard linear mRNA barcode, the MBC, but the second is a constitutively expressed, highly stable circular RNA called a tornado barcode, or OBC.
A circular tornado barcode. So instead of a standard linear sequence that cellular enzymes exonucleases can easily chew up and degrade, the cell is producing a closed loop of RNA that just survives indefinitely in the cellular noise.
Precisely. Because it's circular, the enzymes can't easily grab onto an end to break it down. It acts as an incredibly stable secondary signal.
It's essentially digital two-factor authentication for a cell. To trust the signal, to believe that the enhancer actually fired and it's not just crosstalk or ambient noise, the cell's sequencing data must present both the MBC, which is like your password, and the tornado OBC, which is like the secure text code sent to your phone.
That's the exact mechanism, yeah. If a cell only shows one of the barcodes, the algorithm mathematically flags it as a false positive and just throws the data out. They tested this dual barcode system using two different libraries. A relatively simple single-sequers library with 20 barcode pairs and a highly complex multi-sequers library packed with over 3,600 barcode pairs.
But let's look at the hard truth of the results here. In the highly complex pools, only 10% of the pairs matched perfectly in the cells. Even in the simpler pools, only 50% matched. Now, filtering the data to strictly include only the concordant pairs, the ones with verified two-factor authentication, did improve the specificity, but it still fell significantly short of the gold standard of testing the AAVs one by one.
It did.
Why? Even with a brilliant two-factor circular RNA filter, why does multiplexing still fail to match individual testing?
Because multiplexing inherently forces a massive competition for cellular resources, making it deeply vulnerable to cell abundance and enhancer strength.
What do you mean by competition?
Well, if you have a weak enhancer, or an enhancer designed to target a very rare sparse cell type, its incredibly delicate signal simply gets drowned out. It is biologically overwhelmed by the ambient RNA and the sheer volume of transcripts being pumped out by stronger enhancers in more abundant cells. The biological noise floor is just too high. Error correction can filter out the false positives, sure, but it cannot rescue a true positive signal that is simply too quiet to be heard.
Wow. So bring this all together. What does this actually mean for you, the listener? Because we are constantly bombarded with headlines about big data, massive multiplex screens, and computational biology solving everything. It creates this illusion that we've mapped all the shortcuts.
Right. But this paper proves that the shortcuts are fundamentally flawed right now. Biology is densely, unapologetically chaotic. Multiplexing sounds like the ultimate modern tool for neuroscience and therapeutic discovery. But until we invent entirely new physical mechanisms to stop AAVs from recombining in the factory and stop them from sewing themselves together and crosstalking inside the human nucleus, one-by-one validation remains the painful, necessary, undeniable gold standard. There was no biological free lunch.
The muddy waters are still muddy. Which leads me to a final provocative thought for you to ponder as we wrap up this deep dive. We've been talking about this strictly in the context of screening and research, but let's apply this to clinical medicine. If our viral delivery systems naturally link up and continuously crosstalk inside the brain, how is this going to affect the future of combination gene therapies?
It is arguably the most critical question facing the field right now.
Think about it. If a doctor tries to deliver multiple different genetic cures simultaneously into a patient's brain, say one to stop a neurodegenerative protein and another to boost cellular energy, are those distinct therapies going to stitch themselves together? Could this concatemer crosstalk cause the therapies to misfire, turning a cure into a dangerous, unpredictable bug? Or, if we are incredibly clever, could we intentionally design AAVs that are meant to link up, using that crosstalk as a feature to trigger complex sequence-specific cascades inside the human brain? Are we looking at a biological failure today, or the blueprint for a whole new level of genetic engineering tomorrow? Something for you to think about. Until next time.